Morning. Good morning. We are starting today with something that usually we do it on the first day, some presentation about the participation in this training school. So as you know, we published a call for this training school in July, early July. And we really got a very high number of applications. So this was very good. There were a lot of people interested in this school. So actually, as you see here, we got 46 applications for 30 different organizations, universities, water suppliers, research centers, public health authorities, and so on. Initially, we planned this school for 20 participants here, physical participation. But because we had this high number of applications and because we had a little bit budget left, we were able to raise the number to 30. So we have 30 participants in this school with physical attendance from 15 countries. So the number of countries is very, very good, and coming from 21 different organizations. So in terms of participation, it was very, very successful. Also, as you know, we stream the meeting, the whole meeting, and it is recorded and it will stay there forever in our YouTube channel and also via Zoom. And now some things about what COST association wants us to have in our meetings. COST promotes gender balance, and as always, as in all our meetings and training schools, we have a very good gender balance. Actually, 77% of participants are female, so we are very good with this metric. Also, COST wants us to promote not only gender balance, but also especially to promote young researchers and young scientists in the field. So this here you can see the age distribution. We are very good. Because the subject is the subject of risk assessment needs more expertise, and it's suited also to senior researchers and scientists. The mean age we have here is 44. But we also have young researchers. COST associations considers a young researcher to be below 40 years of age. We have a very good, again, age distribution. Now also COST wants to support the so-called Theodoros can help, inclusiveness target countries. Okay, I remember the term. Inclusiveness target countries are countries that are considered from COST or from the European Union to be less developed, not in the same level regarding science and technology. As you see here, we have a very good participation of these countries, like Greece, Turkey, Serbia, Albania, Bulgaria, Cyprus. All of these are inclusiveness target countries, but and also we have some countries that not in this group, but the majority of countries are inclusiveness target countries. So this was very also good for our meeting. And of course, we want to have in all our meetings and training school, people coming from various organizations, not only universities and research center, but especially for our action, we want to have people from water supplies, as we have here. From water, we have participation from Water Association, from Water Association in Bulgaria. from public health authorities also. And we have also some private sector consultancy, environmental consultancy company. So this is a very good balanced, again, participation in terms of different kind of organizations. And most of our participants are physical here participants have a PhD. This is I think because the subject of this school is a higher level risk assessment. It's not very technical, very specialized, for example, laboratory training school. And the rest, so almost 80% have PhD, and the rest are PhD candidates. So again, balance. A few things about our budget for this meeting. We estimated, and Theodoros can correct me, around the budget will be around 42,000 euro. We never know the exact number before you submit your reimbursement claims. And all of this budget, 100% goes to cover travel expenses, accommodation, and daily allowances of participants. There are no other costs. But of course, to be able to do this, we really want to thank the organizers, Emanuela, Simona, Maura, and the rest of the trainers, Olga, Chiara, and because not only they did a lot of work, as you see these days, to prepare this training school with excellent materials and excellent presentations, but they also were able to offer this fantastic room with all the facilities, the equipment, technical support for free, also coffee, the suites we have, everything is for free, so that we're able to also have a few more participants. Also, as you know, organizing these such meetings and training schools also has a lot of work done behind the scenes before the meeting, during the meeting, and after the meeting, in administration, management, especially the financial part, because we have 30 people filing reimbursement claims that have to be handled and so on. And this is work done in Democritos, so we especially want to thank Theodoros, who is taking care of every detail in a very efficient way, as always. And finally, of course, we are grateful to Cost Association, our funding organization. Hope that also in the future, collaboration and putting another cost action. So this is.
Hi, good morning. And first of all, let me thank Tri and Theodoros for the support in organizing the training school and for giving the possibility to such a nice group to be together in Rome and to exchange a little bit of our expertise, which is apparently not the core business of the people working in Watertop. But at the very end, I think that your take-home message will be that to know a little bit of the procedure that are usually used to derive guidance value, and to understand if the water which is supplied to the consumer is safe or not safe, make the picture for everyone a little bit clearer. And we are not pretending you to become a risk assessor in 3 days, or an expert of in silico methods and the OECD toolbox, but at least you know that they exist, and you know which are more or less the basic concept, and you can go to other colleagues in your countries which are expert in the field, and you know now which question you can pose them, and you know which language they have. So I think this is something important, and I think that at least you get the taste of which are the processes. And since we want to complicate a little bit the situation, during the first day, I introduce you to the risk assessment to single chemicals. And today it will be dedicated to the combined exposure. That means what we call the mixtures toxicity, or sometimes you see even in the newspapers, the cocktail effect, which is a nice visual situation of the combined exposure, because in a cocktail you have different ingredients mixed together, and the mixtures makes the taste of your cocktail a little bit different from the taste of the single components. And this is exactly what can happen with the taste and odor compounds. You have taste and odor, different taste and odor compounds that stay together in the water, and the odor or the taste that you can have at the very end of the mixing situation can be different from the odor that single components can have. So you know that even from what I told you on Monday morning, that the EU chemical legislation is predominantly based on the risk assessment for the individual substances. But this is, let's say, more an exception than a rule, because in reality, we are simultaneously exposed to many different chemicals. And there is also an increasing concern, not only in the scientific community, because we know that these mixtures issues is important. I remember in 1992, we organized a meeting in Bologna of the International Society for the Study of Xenobiotics, and it was on mixtures. So a lot of time passed from that date, but more or less we are at the same level. We progressed just a little bit because it's a very difficult issue. So even for drinking water, you have a mixtures of toxin and mixtures of contaminants which are present, and what to do. So first of all, let's define what a mixture is. It is defined as a combination of two or more chemicals that may, it is not certain, but they may contribute to the effect. And you can have intentional mixtures whenever you manufacture a formulated products for the marketing, like a biocidal product or a plant protection product, or even the mixtures of flavoring agents which are used in food and feed. These are intentional mixtures. You put together the active substance, some co-formulants, some diluents, and at the very end, you have your manufacturing formulated products. You can have unintentional mixtures that originate from a single source. For example, when a plant discharge the byproducts and the disposal of their goods in the environment. And this is a possibility. And then you have what we call coincidental mixtures that originate from multiple sources and through multiple pathways. This means that yesterday night, we had in our stomach coincidental mixtures coming from the different food that we had and from wine, and for someone, even some other beverage and drinks after dinner, and coffee as well. So we have a coincidental mixtures in that case. What it is important to know is the mixtures chemically defined, or it is not characterized, or is characterized at various degree? Because the extent of the characterization of your mixture, it's important for driving you through the approach that should be followed. Because whenever you have fully chemically defined mixtures, then you can assess the risk of your mixtures based on exposure and effect data coming from the components of the mixtures. But if it is poorly defined, you don't know which are the components. In this case, sometimes it's much better to consider the mixture as a single chemical. So it is called the whole mixture. An example here is some extracts that what they call the safe natural extract. Remember that what it is natural, and many of you works with toxins, are not necessarily safe. It is not a synonym. It's a natural product. You can use it. It's a cosmetic coming from something natural. It is not safe by definition. Remember, for a toxicology, this is a must. Natural is not a synonym of safe. We spoke about combined exposure, but sometimes there is a confusion between what is the aggregate exposure and what is the combined exposure. You can have an aggregated exposure when you consider a single chemical to which you can be exposed from different sources. For example, a chemical that can be present in food and in drinking water, and even in air. In the case studies that we have on Monday afternoon, you had different chemicals and you gave different allocation factor to the chemical, because that chemical can be present in your life coming from different sources. And when it is inside your body, the internal dose depends on all the different sources. Once it is in the body cannot distinguish, this is coming from the oral, this is coming from the inhalation. It's internal dose, which is important. So if the exposure is coming from different sources, but occur with the different route of exposure, I mean, the oral one, for example, then in order to understand which is the total exposure, you can sum up, because the route is always the same. But when it is not just oral, but it can be also dermal or also via inhalation, then you cannot just sum up, because the kinetic is different. Whenever you ingested it, it can be can have a different kinetics with respect to the inhalation, and therefore the only way to know which is the internal dose is to understand which is the kinetic behavior. For example, you need to know which is the percentage of absorption coming from the different route of exposure. But this is for one single chemical coming from different source. When you think about the mixture, you define the exposure as combined. This means that a single individual can be exposed to different chemicals from the same route, or even from different route, from different source. So you can have lots of combination altogether. we will dedicate the lectures to the combined one. And in the last decades, many documents have been published and even adopted by international agencies. They refer to mixtures, to cocktail effects, the combined exposure. But I think that's one of the most comprehensive document has been adopted in 2019 by EFSA, and it is called Guidance on Harmonized Methodologies for Human Health, Animal Health, and Ecological Risk Assessment. Therefore, it's a One Health approach. Everything is included, trying to give an harmonized approach to the mixtures toxicology, to the mixture risk assessment. And the document give information on how to deal with the various steps, that is the problem formulation, the exposure assessment, the other identification and characterization, and the risk characterization, which is, as you see, very similar in steps to what I explained for the single. It's addressed both the whole mixture approach when the mixture is treated as a single entity, and I will show you how it can be done, or the component-based approach when the risk of combined exposure is assessed based on what you know about the single components and the level of the relative content in the mixtures. One concept which is really important is to apply the principle of tiering. Having a tiering approach means that you start at a lower tier with a simple and conservative approach or assumption. In this way, you can avoid to lose time and resources. If by using the most conservative approach, you will not have any risk, then you don't need to go deeper and to know more, because even in a very conservative approach, everything is okay. There is no sufficient, let's say, no risk is expected. Let's put it this way. So as you can see here, when, for example, you have a sufficient margin of exposure, then you can conclude that based on the worst case, the very conservative approach, there is no potential risk. But if there is a lot of uncertainty, or you don't reach a sufficient protection, I mean, your margin of exposure is 3 or is 5. It is correct. The level is lower than the reference value that you calculated, so with all these assumptions. But if you are not comfortable with such a small margin of exposure, then you need to refine and go into the higher tier. Or the risk manager can take the decision saying, I'm happy with what you did. I will put anyway some measure in order to limit the exposure. So you can go, let's say, in two different directions. What it is clear is that the higher is the tier and the lower is the uncertainty of your risk assessment, because it means that going at higher tiers, you put together more precise information, and as far as you reach the extremely refined assessments, your uncertainty is going to be lower. And you can use different tiering approach for exposure and other assessment. It doesn't mean that if you stop at tier 0 with the exposure, you have to stop to tier 0 also for the assessment of the hazard. It can be different depending on the kind of information that you have. And this is just to give you some example taken directly from the document. You see here you have 4 different level of tiering, and this is just for the exposure. You can have the occurrence data, the consumption data, and combining together the occurrence, it means the level in water, with the consumption, that means how many liters a day used to consume. That for the water is quite simple, but when you have to deal with food, the situation can be very different. And then you have your exposure estimate. So you can use default values or the permitted values. For the consumption, you can take the 2 liter that WHO used to use, and then you have a semi-quantitative point estimates. If you don't have any risk at this level of exposure, taking the worst case, you don't need to go and refine the exposure estimates. But if you are not happy with your assessment or with this assumption, you have a potential risk, then you can try to refine your assessment, go into tier 1, and then you can not use the default values, but go for and searching for experimental data, or if the experimental data allows you, you can even model the data. And then you can go for the food basket and not having just the default values. In this case, you have an estimate which is deterministic. But if you are not happy, you can go and go for further refinement, and then you get the semi-probabilistic or the probabilistic exposure estimate. So it just depends on which is your goal and which are the results coming from the different tiers. And you can decide it, I will stop here or I will go and try to get other information. One thing that it is really important, I told you, it depends on the purpose of your evaluation. It depends on the question you have to answer. And this is valid for any work in the scientific area. There is not any optimal methodologies that fit for everything. There is optimal methodologies that fit to answer a specific question, but maybe that for another question is not good enough. And this means that you have in front of you what is called the problem formulation, that give you exactly which is your term of reference, which is the question you have to answer. This is important for the single chemical, but it is even more important when you have to deal with complex pictures and with the combined exposure. And this is the representation of a problem formulation that you can have in front of you at the very beginning. In an institution like EFSA, for example, you have the requests coming from the risk managers that ask people in EFSA to answer a question, is this risky or not? That means that a risk assessor needs to give an answer to this question. And sometimes it's also an iterative process, because depending on the results, they can ask you some more, or they can say, it's okay, I'm happy with your answer. And so based on the problem formulation, you have to address first what we call the gatekeeper step. That means ask yourself, is a combined exposure is really important? And so if it is important, which chemicals should we be considered together? It is called the gatekeeper step because it depends on the likelihood that a chemical can co-occur in the scenario that you have to consider. And if the co-occurrence or the co-exposure within a relevant time frame is unlikely to occur, then you don't need to go for a combined risk assessment. I mean, if you are not going to be exposed, it is exactly as for the single chemical. The single chemical can be even highly toxic, but if you are not exposed, the risk is not there. So if there is not the possibility that the two chemicals are together, then you don't need to consider it. For example, for the risk assessment regarding water, those chemicals that are unlikely present in water don't have to be considered necessarily in every risk assessment from drinking water. I mean, the most lipophilic one, like the 1-octane trione that you have that are almost insoluble. You don't have to every time to think, oh, maybe that's they are present as well, because you complicate your life without any meaning, any real sense. So use your brain every time and try to answer this question, which is really important. Is combined exposure assessment warranted? And if yes, try to characterize your mixture. Sometimes the kinetic information are very important because you don't need to have to drink your water contaminated by some chemical at the same time. You can also have the possibility that you have in your body some persistent compounds that are circulating in your body anyway. And they can interact with the contaminants that you are drinking at that moment. So this is something that you should have in mind. So when you have this information available, it's very important. I put it here an example. If the chemical is eliminated very fast and the half-life is 1 hour, then the probability that your internal exposure is going to last for longer, it is very low. And therefore, an internal co-exposure is unlikely. But if you have two persistent compounds that is circulating, then this probability is much higher. So I told you that depending on the characteristics of the characterization of the mixtures, if you have what in REACH are called the UVCB substance of unknown variable composition, which are generally complex reaction product or biological materials, you don't know exactly which are the components. And for this reason, you can use the Wohl mixture approach. So you consider the Wohl mixture as a single chemical, and then you have exactly the same approach that you can have for single chemicals by testing the mixture. So the mixture is tested and the risk assessment is conducted exactly as I told you for the single chemical. But what it is possible is that whenever you have data on similar mixtures, you can still consider to apply the read across. But again, if your mixture is poorly defined, sometimes you don't have the data to compare the mixtures. And if you don't know all the composition, how to deal with that. In some cases, you can identify marker substances within the mixture, which are considered the major component. And if you have indication of this major component, you can try to compare with the others. And you can even try to apply the TTC approach. This is detailed in the guidance document. I don't want to go into the details during the presentation, but just to have an idea that this is possible. The level of exposure is, again, something which can be dealt with exactly as for the single chemical. It is not that different. I mean, the pathway in the environment are more or less the same. And you still have to consider the more relevant exposure scenarios, just combining the occurrence data on chemical with their consumption data. The advantage of using this Wool mixture approach is that it takes an holistic approach. You know exactly which is the effect of the mixtures together, and you already considered the possible interaction between the chemicals which are in the mixtures. But this is the only advantage that you have, because the major limitation is that these mixtures are not stable. They always change over time, their composition. Think about PCBs mixtures like the Arochlor 1254. You probably have listened and heard about it. When they are discharged into the environment, the relative presence of the different variants of the different congener is not stable, because they have a different kinetic of degradation in soil, or in some cases they have a different photodegradation. So in a short period of time, their composition is going to change. And there you test a mixtures which is no more present in the environment, which is going to change and to be unstable. And so the combination that you can have, depending on the different concentration and the different relative ratios between the different components, even having 5 single components is enormous. You cannot test everything. So testing the real world complex mixtures is not feasible, because the huge number of possible combination make it impossible to check for the real toxicity. So this is, let's say, what we call in Italian, we say is the last opportunity that you have. Whenever possible, try not to use the Wool mixture approach, because it requires quite an extensive and costly experimental data collection. So it is much better to use the component-based approach. You can use exposure and hazard data of the individual components, starting from a very simple assumption that using the dose addition model as a default is the preferred option. What does it mean using the dose addition model? Is making the assumption that the chemical in the mixtures act by exerting their effect without interacting with the others, just summing their effect. There is no possibility to increase or decrease the toxicity of the other components. So this is the dose addition model. Don't take into account possible interaction. I mean, what it is called synergism or antagonism. Synergism in when you have more than the addition, and antagonism is that when you have an effect which is less than the addition. And by looking at all the data that we have available, the dose addition has been shown to be applicable to a wide range of endpoints, and it's, let's say, good approximation of what it is observed regarding the combination effects. Because what you can have just having two substances, A and B. A and B can go and be separated, giving rise to individual effects, which are not even similar, and therefore you don't have to sum them up, because they have different target organs, they have different mechanism of action, they have different metabolism, and therefore they act individually. This is what we call the dissimilar action. You see here, when you gave A and B alone, this 10 is the effect. When you gave A plus B or B plus A, then you have exactly the same level. So nothing happens. Dissimilar action. They don't know each other. You can, when the effect can be similar on the same target, on the same target organ, with the same mechanism, on the same molecular target, like the inhibition of phosphatase for microcysteines, for example, then you sum the effects that A and B can have. And if there is synergism, you have an effect which is not 20, but is 30. If you have antagonism, the effect is not 10, is 5. But based on the data we have, at environmentally relevant concentration, synergism and antagonism are very unlikely to occur. So can be concluded that the dose addition is the preferred and considered as the default model that can be used. In the dose addition, the substances are assumed to behave as a simple dilution of each other. So it's like having the same chemical at different concentration. There are methodologies that I will show you in a minute that allow you to go for the dose addition. But why we can conclude that at environmental relevant concentration, we will not take into account synergism and antagonism? Because based on the data we have, we realize that whenever you have different chemicals which are present and at levels which are well below the NOEL, you don't see any interaction. So we have data to say that. It is not just something coming out of the blue. So dose addition is something that you can use. What to do when you have quite a high number of chemicals? The best way is to organize assessment group, putting together those substances that have the same mechanism of action or the same target, so that you know that you can sum them up. Otherwise, you are summing apple and pears altogether. Here in this table, there are some criteria that can be used for grouping chemicals. Grouping is quite an important part of this combined exposure assessment. This is the reason why in 2021, EFSA has adopted another guidance document just to explain how to group. Because the group is really something that can drive you. But even in grouping, you can use the tiering approach. I was telling you, you are summing up apple and pears. This is exactly the tier 0. When you have 10 chemicals present in your drinking water, and you don't know too much about the chemical, the tier 0 is, I will sum up everything together as they were all in the same group. If what I get is something which is not risky, I will stop. I don't have to go further. It is very simple. It is very conservative. If by putting everything together, you can say, my water is supposed to be safe. It is such a conservative approach that you can be sure about. But if you think that there is no sufficient protection, then you have to refine. And when the refinement is needed, you can create a subgroup of chemical based on other criteria, saying, okay, all these are hepatotoxic. I will put them all together and see if by summing them up, the effect on the liver is consistent with the absence of risk or not. So you can use tiering approach for the hazard as well, and for the risk assessment. As I told you, there is this particular document in which some of the chemical are considered going along this decision tree, saying, I have a number of chemical. First of all, I can realize if I know the mechanism of action or the AOP. You see here a very simple representation of what the mode of action is going from the external exposure to the adverse outcome, considering both the kinetic part and the toxicodynamic part, and what an adverse outcome pathway is. I think that then Olga will explain you a little bit better. But let's say that the adverse outcome pathway is the toxicodynamic part of the mode of action. When I say mode of action, it means that also the ADME, the absorption, distribution, metabolism, and excretion is in. For the adverse outcome pathway, it is just what is called the molecular initiating event, the molecular target, which is hit by the contaminants, going on through a cascade of events till the adverse outcome pathway. If they have a common mechanism of action or a common AOP, if the answer is yes, you can include it in the same assessment group. If the answer is no, you can put in two different ones. But if you don't know anything about the mechanism of action, you can consider common toxicological effect, liver effect, for example. Yes, include in the same, no, exclude from the assessment group. And then you can go directly to the common target or organ system. So whenever you don't know, you exclude. And then you go for the risk characterization. You can calculate the ratio of exposure and then identify those components in the assessment group that represent particularly important risk drivers for the component-based approach. Again, for the wool mixture, it's like the single chemical, and then you can use exactly the same procedure that I show you on Monday. But for the others, in which you have to use the component-based approach, how can you sum up everything? The easiest way, the lower tier, is to use these models, which is called Hazard index. Again, there is a lot of confusion because they use, this is the people in the US, they use Hazard index to go to indicate the risk assessment. So they make a lot of confusion with the names, but this is a way to sum up the possible risk coming from the different components. What you do is to sum up the ratio between the concentration and the reference value. I mean, you did the ratio as for any single chemical, and then you sum up. And once the HI, the Hazard index, is higher than 1, then there is a potential risk, and you need to go for refinement. Again, you put everything together at the very beginning, and then you start to consider grouping. The other possibility is to use the relative potency factor and the toxic equivalent factor. You probably heard about the TEF because it has been very popular, especially for dioxin-like compounds, starting from the 2378 TCDD and all the dioxin-like chemical. In this case, you know from the very beginning that the chemical to which you apply the relative potency factor or the toxic equivalent factor has exactly the same molecular target. And then you know that you can put them together in the same group. One of the chemical, which is usually the substance for which you have more data or which is considered the most toxic, is taken as the reference, the index compound. Here I indicated as the substance int as index. And then you calculate the relative potency factor with respect to the substance index. And you adjust the dose or the concentration of the mixtures by summing up the contribution of the single chemical. Theoretically, it's very difficult, but I have some example. I will show you numbers, and then everything will be easier. In this case, for example, you can find a lot of document about the toxicity equivalent factors for dioxin-like compounds. It was present in the EPA. In this case, the index compound is what is called the dioxin, the 2378 TCDD. It is given 1. And all the others have a ratio which is compared to the toxicity of the TCDD poses 1. And they have values which are lower than 1. It is true that you can even use the other models, which is very similar because the theory is the same, the name is different. It has been used for PFAS, for example, by RIVM. There is a document, but I should say that it is really too preliminary, and they took information from the biomonitoring studies. Yeah, I know, but the problem is that there is a big differences between the toxicity of the different variants. I mean, if you take the C8, they have like PFOS and PFOA. They have a totally different behavior with respect to the C4. They have different half-life. They are not as the C4 are not as persistent as the other. They are less toxic. So it seems that they have different mechanisms. Let's say it's a little bit preliminary to use the RPF for the PFAS. But I know that they are using it. I put it as an example, but I think it's a little bit preliminary. This is what the WHO evaluated as TEF. You see, for example, that this congener, this dioxins, has the same toxicity as the TCDD, but all the other as a lower TEF because they are not as toxic as the TCDD. It was done in 2005, and it is based on the binding to the nuclear receptor, which is, let's say, the key event in the toxicity of this compound. So based on the binding, they elaborate this TEF. But since the binding is done in an in vitro context, they don't consider, for example, the possibility, the different kinetics of the different compounds. So in more recent time, it is in 2008, EFSA re-evaluated the dioxin-like PCBs in feed and food. And I should say I disagree with many of the conclusions that they adopted, and it is not just my opinion. Many member states complained because they choose data coming from epidemiological studies, which are not robust enough. But all member states agreed with EFSA, saying that the current WHO TEF coming from 2005 should be reevaluated in order to take into account all the new in vivo and the new in vitro data. And this is due to the fact that especially this PCB 126 is giving results which are not compatible with the TEF. So once you apply a methodology, it is not written in the marble. It is not written on stone. Depending on the information, you can change quite a bit. There is also another example, and there are many examples in the literature. I took these pesticides because pesticides can be in the drinking water as well. All these pesticides are inhibitors of the acetylcholinesterase, so they have the same mode of action, the same target. And so on the basis of that target, they took the acephate as the one. You see that here, when they tested azine-phosphatidylmethyl, it was even more acetylcholinesterase is inhibiting compounds than the index compound, and phenamifos is 13.5. So they took acephate as an index substance, but at the very end, it was not the more toxic. But it is not important that it is the more toxic. What it is important is that the toxicity is referred to one substance, which is taken as one. So the cumulative acute assessment of these selected pesticides was calculated considering that. And the residues concentration for a pesticide was multiplied by this RPF number in order to have at the end an equivalent content of organophosphate pesticides expressed as acephate equivalents. Why are we not considering the interaction? And we are just looking at the additivity. Because in 90% of the case, the interaction is due to kinetic reason. It is not due to the dynamic, which are possible, but are less frequent. And you can understand when you have a deviation from additivity by knowing the kinetic of the compounds. I give you here some example for understanding what interaction can be. Think about the enhancer in your everyday cream that you put in your hands or on your face. Usually the cosmetic ingredients also have some components that increase the absorption of the active substance. Otherwise, your wrinkles will remain always the same. This enhancer increase the absorption of the active substance. Therefore, there is an interaction because you think to have a given absorption to the active substance, but with the other components, you have a higher level of absorption. Or when you block some pathway with pharmaceuticals, because you block the uptake of a chemical by blocking the transporter. So you can have example also in the modification of metabolism by inducing the enzymes which is going to metabolize the substance. But this usually occurs at quite high doses, which are not the environmental one. I put here this, it is Spanish. I should say that I found it, I was in Spain to give a lecture on the argument, but I like this differentiation between synergy and antagonism that you can have with pharmaceuticals, because by default, pharmaceuticals should be active. And you give the pharmaceutical at very high doses, otherwise they are not effective. They are not efficient. So the exposure to pharmaceutical compound is usually high. And this is why you can have this drug-drug interaction, which are quite frequent. And many of the adverse effects caused by some of the chemical can come from the drug-drug interaction, because you think to take that dose of the pill, but then since you take another pill that enhance the absorption, that block the metabolism or whatever, you have actually a very high dose in your blood. And so this is, let's say, sort of guide when you can have possible interaction. Try to answer one of these questions, and if the answer is yes, then you have a possible interaction. Is can one or more components significantly enhance the uptake of the other components? Or inhibit significantly the excretion of the clearance of the other? Or they act through the formation of active metabolites, and they use the same metabolic machinery to produce that metabolites. So if the answer is yes, think about the possibility to have an interaction, to have synergism or antagonism. And then a case studies, just to answer CC question from Monday. So think about microcysteines, the most popular cyanotoxin we know. We always think to MCLR because it's the most studied, but we know that we have more than 2,000. I think that now it's nearly 300 variants known so far. And the different species can produce 3 or 4, usually not more, different analogs, different variants. But you can have also different cyanobacteria, different species that produce different variants. So in raw water, you can have more. And don't forget that microcystin toxicity is variant specific. They have a different toxicity depending on the structure of the variant. So by adopting a conservative approach, I almost finished. So I think that's acute toxicity may be referred to MCLR equivalence. This is what is also indicated in the WHO background document. Considering that all variants have exactly the same toxicity of MCLR. So this is the low tier. I put all the variants together and they are all as toxic as MCLR. And since it is considered the most acutely toxic, it is supposed to be a conservative approach. Just to give you the idea that you can have very different level of acute hepatotoxic potential going from MCLR to MCRR, one single amino acid, the difference, 1 order of magnitude difference in the LD50 IP. And this is due mainly again to toxicokinetic reason, because when you look at the ability of the different variants to inhibit phosphatase, the level of inhibition is more or less the same for all the variants, but they have a different possibility to enter the cells due to transporters, because the affinity for transporters is different, and they are differently metabolized by glutathione S transferase. So there are differences among variants, and there are differences even among species. This is the mechanism. As you see here, the molecular initiating event is the inhibition of phosphatase. So it is correct to put everything together. But you can see that for the acute toxicity, you have a cascade of events that lead to intrahepatic hemorrhagic areas. While if you go for repeated toxicity, you go for cellular proliferation, is a totally different cascade of events. So you cannot mix the two. So we said that by adopting a conservative approach, I can put everything together. And this is an example. So I have these mixtures. I just make the sum here. And this is something which is not extremely robust, because to understand which is the acute risk is a little bit tricky. And the trickiness of the problem is that you have a very steep dose response curve. But just to have an idea, we can identify this acute no effect dose of 2.5 microgram per kilogram body weight, which is 150 microgram for an adult. So if you consider that you can have, you can consume 2 liters a day, the intake is 450 microgram. This means that you exceeded it. Why? I don't know why I put it no acute risk is expected, but so forget it. This was probably a copy and paste, but the risk is there. So since the mechanism of action is the same, you can apply exactly what you can apply to the toxicity equivalent factor, exactly as we did for the TCDD. So the TEF is applied, and you see here that if you compare the IP LD50, you can have this kind of TEF, and therefore the example is give you the idea that instead of having 2025, you have 77, which is a little bit different. So by applying the TEF, your acute toxicity, the risk for acute toxicity is greatly reduced. Don't forget that this is just an example. You still have limitation. The data are from IP administration. This is not the normal way you are exposed to. There is a big difference between the LD50 if you go for the IP or if you go for the oral due to kinetic differences. So let's say that this is not extremely solid, but you can do it. If you think to the repeated exposure, you don't have data. You have just data for MCLR. You don't have data for the others, and you cannot extrapolate because the pathway are totally different. What you see with the acute to what can happen with the repeated toxicity. This is exactly what we discussed on Monday for the MCLR. Again, for the chronic toxicity, you consider that all the MC present are equivalent to MCLR. This is a very uncertain assumption because we don't know exactly which is the repeated toxicity for the other variants. So we are extrapolating, but we have a lot of uncertainty in this extrapolation. And for the aggregate, consider also the possibility to have microcysteines in food. I think that I can stop here because then what I want to show you is that we can say something, but we have hundreds of open questions when you think about mixtures. Do we have enough information about the co-occurrence of, for example, mixture of toxins in reality? Can have information on the mixtures of other contaminants, persistent, not persistent, organic, inorganic? What about the mixtures of toxin with the other contaminants? And if yes, in case we say, oh, we have the information, then I'm posing you other question. Is the database for the toxicological properties of toxin able to support a combined risk assessment? At the moment, my answer is no, because we don't know exactly what happened in the repeated exposure. Can we group different toxins based on their mode of action? Apparently, for some of them, the answer is yes. Can we identify the data gap and try to prioritize them? So, I mean, I think that's what we need is start to answer this question, and otherwise we will still remain in this middle situation in which the picture is not that clear for us as well. Thank you. For the people online, I think that we are going to have a 15 minutes break. And so if you have any question, you can ask me in front of a coffee.
Yes. Yes. Okay. Okay, good morning, everybody. And welcome to the last presentation of this course. And today I'm going to tell you about adverse outcome pathways. The abbreviation is AOP. I know that some of you are using the same abbreviation for different things, but this one is for adverse outcome pathways, and the title of my presentation will be AOPs from research to regulation. So what are adverse outcome pathways and how this term has been created? We should go back to 2012 when Organization for Economic Cooperation and Development, OECD, started this AOP development program, and the responsibility on development of this program was under EGMS group, which is the extended advisory group of molecular screening and toxicogenomics. We are the member of this group from 2014, so we were able to follow all development of this program at the OECD level. So it was introduced in order to try to improve the use of mechanistic application information in different application of predictive toxicology in order to increase the confidence of predictions. So this just a very short background, you know that there are many untested chemicals on the market, not fully tested chemicals, and in vivo tests are very time and resource consuming. So it's necessary to try to develop the methods alternative to animal testing, alternative to in vivo testing. And also to increase their confidence and facilitate their regulatory acceptance. So at the level of OECD, we have already several OECD test guidelines based on non-animal approaches. For example, for skin and eye corrosion, irritation, phototoxicity, genotoxicity, skin sensitization, and they're very important because OECD testing guidelines contain already validated methods. And as has been told also several times during the previous 2 days, it's very important to have high quality data, in this case also supported by mechanistic understanding, in the case of application of AOP, just to be able to predict with high level of reliability the toxicity property of target chemicals with unknown toxicological properties, starting from the different properties of the analog substances, using, for instance, Rita-Cross or quantitative structure activity relationship QSAR models. So just some more wide background. There is here you see the citation from Sidney Brenner, who was a Nobel Prize in molecular biology. And he said that a crisis developing in biology because we have completely unstructured information, which does not enhance understanding. So he underlined the necessity to create a framework to accommodate all this biological unstructured, not very well structured, at least, information. So let's start from the explaining of the structure of an AOP, of an adverse outcome pathway. You see here different steps in this stair. They correspond to different biological levels, and each AOP always starts from molecular initiating event, which is MIE here. So it could be, for instance, protein binding of a chemical or DNA binding or something else, receptor activation. Many different events could start the whole pathway. And each pathway contains different key events. Each step on the stair corresponds to one key event at different level of biological organization. You see cellular, tissue, organ level, then coming to the organism level, and finally to the adverse outcome, AO, where we observe a toxicological effect on the organism or population. So there are this sequence of key events, of which all of them are related to each other. We will talk about key event relationship in the next slides. And then finally, this pathway arrives to the adverse outcome. So to explain you using an example, this is an AOP, one of the first AOP endorsed by OECD. It's AOP on covalent protein binding leading to skin sensitization. And here we see different key events, and on the MEIM, it's molecular initiating event. Here is protein binding. That could be also confirmed by in silico models quite well. Then there is protein binding in chemical and different responses at cellular, organ, and organism level. And here several key events, they have different nodes, and each node corresponds to one of the available in vitro assay, which is available. It could confirm or not confirm the key event, if a key event occurred or not. So this is another example on aromatase inhibition leading to the reproductive dysfunction in fish. And so here you see different key events starting. It's just a screenshot from the AOP it has been endorsed by OECD, by EGMST group. And you see that there are different key events starting. The initial event here is aromatase inhibition that directly leads to beta-estradiol synthesis reduction, then plasma reduction of plasma concentration of beta-estradiol, and then reduction of vitellogenin synthesis in liver, and then again directly leads to the next key event, which is reduction of plasma vitellogenin concentration. And then there is a damage to the reproductive system in fish. So this is the final adverse outcome. And in this table, you see also different column. This one, for instance, is weight of evidence, because when an applicant submits an AOP to OECD for consideration and eventually for endorsement, it's necessary to present also different explanation for each key event, for key event relationship. We will also talk about this later, but it's important to support the AOP by expert judgment on weight of evidence for each key event and their relationship. And also, if possible, the quantitative understanding is also important, even if we have several, let's say many AOPs which are just qualitative, not quantitative, but again, I will explain about this a little bit in more detail later in my presentation. This is, let's see the AOP of aromatase inhibition and dysfunction of reproductive system in fish in this using this animation. This is a normal egg production in fish. Everything is working well from aromatase enzyme to the egg production. But here in the next slide, we have one chemical. It's important to say, maybe I forgot to mention it before, that AOP are not chemical specific. They are not considered to be chemical specific. They are just biological pathways consisting of the sequence of key events and their relationship. But then, of course, to start, in order to activate the pathway, we need a chemical stressor. And here we have this fadrozole, which is endocrine disruptor. You probably know that endocrine disruptors are the class of chemicals that are able to damage the hormonal system of human and animal organisms. So we have this endocrine disruptor in action, and which inhibits the aromatase enzyme here and reduce estradiol synthesis and its concentration in plasma, and then reduce vitellogenin production, and then reduce also vitellogenin uptake in ovary in female fish. And then we see the significant population reduction. This is the whole pathway that starts from the initial molecular event caused by chemical stressors, in this case by endocrine disruptor fadrozole, coming to the final adverse outcome, which is population reduction in fish. So using the schema that I have shown you in my first slide, here you see the mapping of key events on this different level of biological organization, which start from enzyme inhibition, from initial molecular event, and then coming to the population reduction in fish in vivo. So why it's important to have this AOP and understanding of adverse outcome pathways? If we have a target chemical without toxicological experimental data, and then we have another chemical with different data coming not only for adverse outcome, not only on the final toxicological effect, but also on the intermediate step. We have also some in vitro tests also validated, sometimes even validated by OECD tests, which are available for the key events. And so we can increase significantly the confidence and reliability of our prediction if we will use this prediction not only on the level of the final adverse outcome, but also on the level of when possible, where we have the data for the chemical with known toxicological property, also at intermediate levels corresponding to different key events and different level of the biological organization. And here, okay, it's also important because not and the dose effect concentration is dose effect relationship is important also for the AOPs. For instance, sometimes the concentration of chemical stressors is not enough to activate the whole pathway. But here you see another example. You see that one chemical stressor can activate different biological pathways. It's happens almost always. So it's not only one biological pathway, but many biological pathways that may be activated by the same chemical stressors. And let's see here at first, you see on this animation that all pathways have been activated by this one chemical stressors, and all of them arrive to the final adverse outcomes. So what's about mixtures? It's difficult always to evaluate the toxicity of mixtures, and as you understood from the presentation of Emanuela from today morning, but sometimes it's just an illustration that sometimes two chemicals can share the same key event, and then, for instance, they activate different molecular initiating events, but then the pathways, they have the joint pathways after a certain key event. For example, we see here a key event, a common key event for two different chemical stressors, and it might be also a mixture. And this way, if you start, for instance, the first chemical stressors, it might happen that the concentration of the first chemical is not enough to activate the whole pathway, and it just stops and key event here, and does not cause any adverse outcome or toxicological effect. But when in the mixture we have another chemical, another component that starting from totally different molecular initiating event, at certain point comes to the same key event, you see that also the rest of pathway is activated. And we will observe here the final adverse outcome. So, as you understood, our piece are the way of organizing of biological information, and we have a lot of information, existing information. And the most important goal of the RP developers is organize all this information in structured way to make it helpful and useful to RP consumers. Or who are RP consumers? Of course, there are different type of stakeholders: scientists, toxicologists, physiologists, also, and of course, and the final step, the regulators, because we need to increase the confidence of the methods alternative to animal testing and to facilitate the regulatory acceptance. This is the final goal of everything. So it's important for developing RP to use high quality ingredients. So what has been said several times in two previous days, that for any methods of predictive toxicology is extremely important to use the high quality data. So how we can construct an RP? We should start from some data extracted from scientific literature, reviews. All source of information should be transparent and help us to understand how the whole pathways, how it happens that starting from molecular initiating event, this pathways arrives to the final adverse outcome. So very often, RP development starts from systematic literature review. And it's a long and quite time-consuming, resource-consuming process. So it's necessary to define very well a scientific question, then to define strategy, criteria for inclusion and exclusion of several literature sources, to extract the right sources, and then evaluate study quality case by case, and extract all necessary data, and also be able to interpret the results, not for the whole pathway, but for all components, starting from molecular initiating event, passing all key events and their relationship, and arriving to the final outcome. So normally it's not almost always a development on one AOP requires a team of collaborators with different expertise. It's usually not a single person who develops an AOP from the beginning. Normally it's a team of collaborators with expertise in molecular biology, physiology, and also risk assessors, and then the ecologists, cellular biology. So because it's necessary to examine different effects at different level of biological organization. And now let's talk about the repository where you can find all existing AOPs. It's the name is AOP Wiki. It's community resources developed by OECD together with Joint Research Center, GRC. And it's a community resource, so it's available to everyone. You can just open AOP Wiki and see different tabs here: AOPs, key event relationships, chemical stressors, developers handbook. Handbook is very important, has been developed by OECD, and there are very detailed instruction how about the best practices, let's say, in AOP development. So here you can also register if you want, but it's not necessary. You can just browse the existing information. Of course, if you would like to submit a new AOP or new key event or key event relationship, any new information, you need to have an account in this AOP Wiki. So there are 3 levels: just guests, then registered users, and then there are some group having the right access, because now there are a group of AOP coaches established by OECD. And for instance, I also have right access. I can even delete the AOP from there, but I will never use this possibility, of course. But there are just a few, not a few, but maybe 30, 50 people having the right access there, beside of administrators. And also another community resource has been created by OECD and Joint Research Center, GRC, which is AOP knowledge base. So it's just a combination of different resources publicly available to everyone. It's AOP Wiki, Effectopedia. It's a resource where you can create to draw some pathways. It's not very user-friendly, unfortunately, from my point of view, but it can be also useful. There is AOP Explorer and intermediate effect database. All this is AOP knowledge base and based on the ontology and control terminology. And we were involved also in the development of this terminology for AOP knowledge base. And yes, and here you see this more or less the same structure presented in different way. This is AOP Wiki, Effectopedia, AOP database, and AOP Explorer and intermediate effect database. Now also intermediate effects database recently has been implemented also as one of the OECD harmonized templates. OECD harmonized templates, they are templates that are developed by OECD in order to standardize the toxicological data, and OECD harmonized templates are available for different toxicological endpoints. And now dedicated harmonized template is also available for the intermediate effect that might be used for AOP development. Then another point, if a group of scientists would like to develop an NLP, or maybe they already have some scientific publication related to the mode of action or something like this, and they would like to develop an NLP that would be officially endorsed, because on NLP Wiki you can also add an NLP that are not part of OECD NLP development program. But if you would like that your AOP, if you wish to include your AOP in the OECD development program, it's necessary to present a project proposal. It's twice per year. It's necessary to fill in a template and present your proposal to the OECD EGMS group. Now it's a different group. Now its name is ASK Emerging Science and Chemical Assessment, and it's necessary to present a short proposal for evaluation. And if the members of OECD, we are also the member representative in this group. If the expert group decides that AOP is relevant, also not only from scientific, but also from a regulatory point of view, normally it's accepted and it's included in the OECD AOP development program. So then before 2019, it was a process, internal review process. Here you see, for example, in AOP, I was one of the reviewers for this AOP that has been successfully endorsed by OECD. So it was after the developers of an AOP were ready to submit the AOP for internal review. It was an internal review process, and then the OECD administrators, the OECD Secretariat organized also an external review process from the expert in the certain toxicological domain to validate also to scientific value of an AOP. So to get one AOP validated by OECD, not validated, it's not a validation, endorsed in the right word. It's quite difficult. And so from 2019, there is a coach group, AOP coaches group at OECD, and we are also the members of this group. So for each AOP included in the OECD AOP development program, from the beginning, OECD assigns one coach who is an expert, who is normally a member of OECD expert group, and who assists to AOP developers during the whole process of the AOP development. So it's also, I think it's a good initiative introduced by OECD in the last years. And yes, to submit and to endorse one AOP by OECD, I believe it's much more difficult than just to write a scientific publication, because you need to fill in many documents on AOP page, then all information on molecular initiating event, which is description, how it could be measured or detected, taxonomic applicability, evidence for chemical initiation, for the same for each key event page. And key event relationship, it's also very important, because here it's necessary for each key event relationship, it's necessary to describe biological plausibility, empirical support, possible inconsistencies, uncertainties, quantitative understanding if possible, and then the same for the adverse outcome. So it's quite difficult process for the AOP developers. And in fact, here for supporting of each these pages dedicated to, especially to key event relationship, OECD suggests to use Bradford-Hill criteria for stable causation. It's essentiality, let's say, to support essentiality of a key event, because why certain key events is important in the whole pathway. It's here it's necessary to document consistency of the evidence, temporal relationship between different key events, biological plausibility, dose response relationship, consideration of alternative explanation. So there are many things that should be included in the AOP description. So normally AOP, the OECD AOP program strongly supports collaborative AOP development. So AOP are modular and they are living document. Also, it's very important to support, and from OECD point of view, it's also very important to support the development of AOP networks. So it means, as it was also one of my previous slides, when different pathways can share, like here, some key event in common, and each pathway can start also from different molecular initiating events, come here, and arrive to different adverse outcome. But there are also some key event in common. So it's very important for biological understanding, also for many pathologies, to understand the whole pathway and the network, different pathway that can be activated by different chemical stressors and the common parts of the pathways. So what we have now, AOP, they are living document, but also because it's quite difficult to get an AOP endorsed by OECD. We have this situation, we have many, in the starting point, we have many AOPs that are not in the OECD working program. That we have the AOPs including in OECD working program, but many of them are there for many years, and many of them have been abandoned by the developers because maybe they don't have time, they have different projects, so they are still there, but they are not complete. They have some documentation for some key events, some key event relationship, maybe for molecular initiating events, but the documentation is not complete. And then we have some AOP that are under evaluation at OECD. And finally, we have the highest level of the AOPs that have been endorsed, adopted by OECD. Endorsement doesn't mean validation, doesn't mean automatic regulatory acceptance. They're just endorsed by following the external and internal review steps, and they are published under OECD development program as a separate documents. And of course, if you would like to use an endorsed AOP in your risk assessment, it's the confidence of your results of your prediction will be higher in comparison if you would use not endorsed AOP. But it's not forbidden at all to use also in regulatory application the AOP that have not been endorsed yet, or even are not in the OECD or AOP development program. It's possible to use them, for example, on the integrated risk assessment, but it's necessary to document very well each step, each key events and key event relationship. So here it's just because this group at OECD, EGMS group, which is responsible for OECD RP development program, there are several experts nominated by different OECD member country. We are also there. And for example, this is an example that a presentation that Italy presented by Italy several years ago, and that underlined the importance, for example, some RP development in the area of non-genotoxic carcinogen, because non-genotoxic carcinogenicity, it's a toxicity endpoint that could be initiated by different mechanisms. So it's very important to understand a little bit more, and ideally it would be very useful to have some AOP in this area, but it's quite difficult, as I said before, because the development of one high quality AOP, complete AOP, it's a long time and resource consuming process. So when a scientific group starts to develop an AOP, it's always necessary and it's extremely important not to start from scratch, but go to the AOP wiki and to see the all existing information, because it's possible that the key events of the key events of the given AOP already there, and they belong also to other adverse outcome pathways. The same for key event relationship, and so it's very important to not to avoid the duplication of key events, key event relationship, molecular initiating event, adverse outcome. If you can find already that it's in AOP wiki, it's necessary to use the same terminology and not create similar key events, maybe using the synonyms. It's strongly recommended to use already existing one, because it's extremely important for the creation of AOP networks. So this is example that construction of AOP network. Again, there are different AOP from OECD AOP program. Then them and all of them started from different molecular initiating events. Then they have also some different early key events, and then they arrive to the shared, multiply shared key events and shared adverse outcome, which are all in common. But the initial part of each pathway, initial parts are all different here. And that's why it's important to create such a network. For instance, there are several projects funded recently by different authorities, such as, for instance, EFSA recently funded a project on the AOP network for metabolic syndromes, for different metabolic pathology like obesity or diabetes. So they also could share the same key events, but starting by different chemical stressors and by different molecular initiating events. So here it's also it's possible to combine. It's totally possible, and it's feasible to combine in the AOP network some the efforts coming from different authors. For example, it's possible to use not completed AOP from AOP wiki, to use already created key events that with the page completed by other authors, and use them in your AOP network. It's also the recommendation of OECD to do so. And the final goal will be to generate new knowledge. And already we have in our wiki more than 9,000 unique paths from molecular initiating event to AOP. Not all of them are endorsed, of course, but the final goal is to create a very wide and OP network where all shared common key events are connected between each other. So what are the most important benefits of IOP? It's effective use of existing knowledge, opportunity for collaboration between different scientific groups, useful in prioritization and risk assessment, development of predictive tools, and the reduce of in vivo experiments, of course. But let's talk a little bit about key event relationship, because normally in AOP, when we have several key events, we have this key event relationship between upstream key event and downstream key event. And so it's necessary to document the evidences, why they are linked between each other. So the OP authors, developers should document the causal evidence, provide weight of evidence evaluation. And so it's quite, again, we go here to the Bradford-Hill criteria, which is very important to document. Bradford-Hill, he developed causality associations for epidemiological studies, but this causality approach is widely used also for the AOP development. It's important to document and provide the evidence of essentiality of each key event in the pathways. So coming here to the next slide, you see OECD, it's required by OECD AOP development program to produce several evidence for key event relationships, such as biological plausibility, of course, and essentiality of each key event in the AOP, the whole pathway, and also empirical support for key event relationship, and where is possible the quantitative understanding. Again, some AOPs are just qualitative, and they probably will remain only qualitative AOP, but also the quantitative understanding is important. And so in the next slide, I will just go a little bit in more detail on the quantitative relationship in the AOP. So normally we have upstream and downstream event, but not always there is some kind of this tipping point, and we don't know at which condition of exposure by chemical stressors this tipping point can be surpassed or not surpassed. So not always the pathway is activated, the whole pathway is activated, because it's not possible to surpass this tipping point and go from the previous key event to the next one. So here, as a quantitative understanding for an AOP, it's necessary to produce some documentation on response-response relationship. So response-response relationship between response of the upstream event and downstream event in the pathway. So coming back to our aromatase inhibition and decrease of reproductive function in fish, AOP, reproductive dysfunction. Here we see two key events from even not really key events, because aromatase inhibition is a molecular initiating event here. And there is a relationship between aromatase inhibition and plasma concentration of estradiol. So this is response-response relationship here, and there is an equation. So in this AOP, we have this also relationship, quantitative relationship for other key event. events here, B and C. So this AOP is fully quantitative AOP. So we have also, we can see if finally adverse outcome can occur or no. We have more information, but also qualitative AOP could be important in predictive toxicology because of course, we don't have quantitative support for all AOPs, even among the endorsed AOP in the OECD AOP development program. So now just let me talk a little bit about the regulatory application of AOP, because what I have shown you before, it's more or less about some scientific background for the AOP development, but how we can use them in some regulatory applications, so to facilitate the regulatory acceptance of alternative methods in modern toxicology. So how we can pass this bridge, how we can construct this bridge between scientific development of AOP and regulatory application. So let's talk about a little bit about IATA. IATA, integrated approach to testing and assessment. It's quite new term introduced by OECD in 2015, I think. And there is also an OECD group on IATA case study project. We are also the members of this group. So the term of IATA has been introduced by OECD as a pragmatic approach, which this is the formal definition by OECD. It's a structured approach that strategically integrates and weights all relevant data to inform regulatory decisions regarding potential hazard and or risk and or the need for further targeted testing, and therefore optimizing and potentially reducing the number of tests that need to be conducted. So explaining in simple word, in one IATA, it's necessary to integrate all available source of information and then applying our expert judgment and the weight of evidence approach, uncertainty evaluation for each source of information, we can come to the final conclusion, for example, on the group of chemical or of chemical without toxicological data based on all available information. But here it's important, the expert judgment. So there is also a guidance of OECD how the AOPs could be used within IATA approach. So in IATA, we use all sorts of information, also toxicokinetics, exposure, all existing information, in vivo, in vitro, in silico, everything. But if we use the information not only for the adverse outcome, maybe for initial molecular event, but also some tests and some information, some experimental information available for the key events, it significantly increases the confidence and reliability of the final conclusion. So also it's important to transparently report all studies, all information used under IATA, to report everything and evaluate the final uncertainty. At first, the uncertainty of each separate source of information, and then also the final uncertainty of prediction made by IATA. So this is just a picture from our article on the genotoxicity in silico prediction. And here it's you can see how the whole picture of the methods that could be used in silico prediction, in silico methods. So starting, it's for genotoxicity, but it's perfectly applicable also for other endpoints. And here there is, you see in the center, the AOPs. that what is the role of AOP here? AOP can inform chemical grouping and data gap filling by read-across trend analysis, and also provide an opportunity to group chemicals, not only based on their chemical properties, but also based on their biological activity in different level of biological organization. And all this IATA integrated approach for testing and assessment can provide a more robust support framework for accessing final toxicological outcome for untested chemical. And also it can significantly improve predictive performance on in silico methods. It's important that each source of information based on it's transparent. And here, if we use AOP, it's mechanistically based. It's mechanistically based data integration, which is more robust from point of view also the regulators, because if you produce some mechanistic support, it's always very appreciated by regulatory agency, especially when you are using an alternative methods. So, but always in the end, there could be different source of information, but always in the end, it's necessary to evaluate uncertainty, provide weight of evidence support and analysis, and there is a final expert judgment that could be also different depending on the different experts and different scientific, different groups of toxicologists. So the final goal is the regulatory acceptance, which is not always possible, but we have some successful example of use of IATA that I will talk a little bit later. But now, for example, also for QSAR model, this is a screenshot for the very recent guidance QSAR assessment framework. And our group is very proud to be part of this this project. We coordinated, we all three, Cecilia, Chiara, and I, we co-coordinated this project for the last 3 years. And together with ECHA, which is the European Chemical Agency. And finally, just a few weeks ago, this testing guidance of OECD has been published. And you see also for QSAR model is the guidance state clearly that when possible, a mechanistic interpretation based on toxicokinetics or toxicodynamics, this case AOP, it's very important when possible. There were also some discussion within the expert group because during the last 2 years, even more, almost 3 years, we co-coordinated this project, but it was an expert group that consisted of more than 40 experts nominated by different OECD member country and regulatory authorities such as EFSA, ECHA, GRC, and not only European countries, but also United States, Canada, or Japan, Australia, so OECD member countries. And there was some discussion to remove this phrase if possible and to make this mandatory, but it was maybe too much to ask for all QSAR, even for statistical models, to provide always a mechanistic interpretation, in many cases AOP based. So we left this if possible, but it's really increases very much the final confidence of QSAR prediction. So now let's talk a little bit about defined approach. What is the difference between, because in IATA we have this expert judgment in the end, and defined approach, it's the same things more or less as IATA, because it integrates all available information, but it's under OECD testing guideline, and so there is defined interpretation of data, fixed data interpretation, and there is no expert judgment inside defined approach. So one of it's the first case of acceptance of OECD under OECD at the OECD level of defined approach is defined approach for skin sensitization. Here you see a screenshot of the testing guideline of OECD published in June 2023. And this defined approach of skin sensitization. It's some kind of YATA with fixed data interpretation. So it includes all sorts of information. And it's important to say that it's very often based on the validated in vitro test, new approach methodologies, but the reliability of the methods, let's say half in silico, half in vitro, it's not less than reliability of in vivo tests. Here you see the local lymph node assay, and also there is Guinea-pig maximization test. There are two traditional in vivo tests for skin sensitization, but the reliability of a defined approach is not it's the same level as the reliability of in vivo study, and this defined approach is totally animal free. So also, let's say just a few words at the end about AOP implementation and the OECD QSAR toolbox without demonstration today, but coming back to the skin sensitization, we have different in vitro tests, which are tests to confirm the key events in the skin sensitization adverse outcome pathways. So we have all these in vitro tests that have been validated by OECD and published by OECD. So it's facilitate enormously the development, the application of this adverse outcome.
Kadın sesini duyamıyorum.